I'm happy, so happy to bring brine flies to the conversation and their other fly friends. I am the facilitator today. My name is Baxter. I have two announcements before we get into the panel. The first one is, oh yeah, there it is, those guys. The first one is that the Great Salt Lake license plate has its final design and is now being printed, and we would love to invite everyone in this room to get a license plate. That's the Venmo if you want to take a picture of that QR code. There's also forms on the check-in table out here if you'd like to. The other announcement is that many of you heard this statement yesterday, and I'm sure almost everyone in this room agrees with it. So if you agree with it, I invite you to send your comments to Representative Snyder. There's his contact information. Tell him how much you agree with these words. There wasn't enough room to put his whole title on here, so I had to abbreviate it. Well, he insulted me. All right, let's get on to flies. Okay. So we have a distinguished panel of fly experts for you today. Dr. David Herbst is here. He is with the University of California and is a free-range biologist working at all the saline lakes in the West, and so we're happy to have him. Carly Biedul, who is Great Salt Lake Institute's coordinator until tomorrow, till the end of tomorrow. And she's with Westminster University with me. Heidi Hoven is the Gillmor Sanctuary, what is your title now? Senior Manager at Gillmor for National Audubon, and she gets to see lots of different flies in the wetlands. Amanda Barth is the Rare Insect Conservation Coordinator at USU, so she's going to talk about some really interesting possibilities for the fly. So I will move forward with just a couple slides of my own. This is from my, we like to say we're related by some polygamous great-grandfather, but we're really not. Scott Baxter, my dear friend, he took some amazing foul rope pictures with flies. There are a number of really important birds that depend on flies at different stages of their life cycle on the open water of the lake. And so for those flies, for those birds, these are in great numbers at Great Salt Lake, the ear-grebe, California gull, both phalaropes, and common goldeneye, which are, of course, diving ducks. So it's not just, a couple birds that eat these. There are great numbers of birds that eat flies. And because I study what they eat, I did want to call out microbialites, where the larvae crawl around and pupate, and they're eating these mats on the bottom, and I want to point to the significance of the microbes in the ecosystem. But we're going to hear more about these different life cycle stages, so I thought I'd just throw this up there to point out that these guys live most of their life underwater, and they only live as flies for a few days. So really, they can be a larva for 6 to 9 months in the lake, which is pretty amazing. So first, let's start with Dave. Dave has a few slides, and do we have a clicker up here? We do. You want a clicker? Okay. And so each person is going to show a few slides and give you an introduction, and then we're going to have a nice conversation, question-answer session at the end.
Let me first thank Bonnie for engaging me in work out here at Great Salt Lake over the past year and a half. And Lynn for inviting me to the forum once again. Last time I was here, I think we had the International Society for Salt Lake Research and a meeting along those lines. Great occasion, but it had nowhere near this number of people. It's as low as I can go. So being in the business of studying brine flies for I think longer than anyone else that I know of that hasn't quit the business, I guess that makes me Lord of the Flies, but I'm an official Salt Lake junkie. I love Salt Lakes all over the place. And amongst them are lakes that are on this map here. So I wanted to start off with a bit of history and geographic context to give you some idea of what conditions are like across the Great Basin and have been. So this is a map that shows you what lakes were like back in the at the high stand, the pluvial lakes, Pleistocene lakes when they were fed by glaciers, and then what they look like after a period of climatic aridity and warming, drying down to the remnant lakes that exist here now. So this includes, see, this has got a pointer on it as well. Very good. So this includes, of course, Great Salt Lake over here. Out here in the western part of the Great Basin, we have Owens Lake down here, Mono Lake here, Avert Lake up here, along with Summer Lake in south central Oregon, and Pyramid Lake is a remnant, Walker Lake is a remnant. I'm not sure I'm pointing to exactly the right spots, but the gigantic Lake Bonneville dried down to Great Salt Lake, and gigantic multi-armed Lake Lahontan dried down to lakes like Pyramid Lake, Winnemucca Lake, and Walker Lake. What's interesting about these lakes is that across the geography of the Great Basin, there's also a lot of geological and geochemical variation. So the lakes that are in the western part of the Great Basin tend to be alkaline carbonate lakes. So high pH lakes that have high concentrations of carbonate and bicarbonate in them, whereas lakes in the eastern part of the Great Basin tend to be chloride-dominated lakes. So along with variation in salinity amongst these different lakes because of their differences in the amount of water that flows into them and their history and their morphological structure, we also have a lot of variation in the water chemistry. And so this has formed a great template against which the evolution of different brine fly species has occurred. And so to give you some idea of what brine flies look like, here's an example. This is from the alkali fly. The alkali fly is a name coined for the species Ephedra hyans that lives in the alkaline bodies of water, primarily again in the western part of the Great Basin, where all the alkaline lakes, most of the alkaline lakes are. And so on the top here, you see on the left, this is what the larva looks like. On the left side is a respiratory siphon. The respiratory siphon in many cases is no longer a functional feature. It's actually just a thinning of the cuticle across which gas exchange occurs. But it does indicate that at one point in the evolutionary history of these particular insects, they were breathing air. These were little spiracles on the tips of those respiratory siphons that could be put up to the water surface again, through which that they would breathe air. And there are many aquatic insects, especially in the Diptera, the fly family, that still use those kind of respiratory siphons to respire, but no longer in the Salt Lake versions. And then below is the pupa stage, and the pupa stage is basically just a hardened version of the last larval skin. So there's 3 larval instars. That means they go through a molt from the first to the second, and a molt from the second to the third, and then in the third molt, they actually turn into the pupa stage. So it's within the pupa stage that the adult fly begins to develop. And from the pupa, the fly emerges, popping off a little hatch cover-like structure at the front of the puparium. The fly emerges and floats up to the water surface in a bubble of air. And at the surface, they take about 5 minutes for them to basically harden the outside structure, expand their wings, harden their wings, and then they can fly off. And down here on the right, at the low end here on the left side is where you see what an adult fly looks like. And you can see they're very hairy flies. In the case of the alkali fly, they're not only very hairy over the surface of the body, but they have a very waxy surface. So they can actually use this to their advantage to crawl underwater. If there's any kind of a substrate rock that's only partially submerged in the lake, they can pull themselves down through the air-water interface, crawl around on that rock underwater, where they can graze on algae or lay their eggs, which is the primary function of that particular behavior. So that waxiness and hairiness is an important attribute, at least of the alkali fly. And here on the right, you can see they occur in really great abundance, great aggregations, and these aggregations really make them an important and easy to feed on food source for birds because they aggregate along lake shores in great numbers. Birds can basically run along, snapping their bill to pick up that as a food source. But birds are also feeding on open water in near-shore areas where they will feed on the fly larvae and sometimes on the pupae. You especially see this behavior in phalaropes. If you've ever seen phalaropes, especially in relatively shallow water, they can spin around. They create a little vortex of water, And at the bottom of that vortex of water, like a little tornado of water, they can pull up any larvae or pupae that might be on the lake bottom, and then they peck at the inside of that vortex to feed on the fly larvae and pupae that they will have pulled up. So one other really important attribute of salt lakes as habitat for the flies, not only is that they are living in an environment that they and very few other organisms can be adapted to, these very high saline lakes, they also require, like a rocky intertidal organism would, a rocky substrate on which they can protect themselves, attach and protect themselves. So it's in that pupa stage where, as you might have seen in that last slide, the last larval proleg is enlarged, and they use that as a clamp. They clamp it down on whatever kind of substrate is available to them. Typically, it's going to be this rocky intertidal type of habitat. Microbialites at Great Salt Lake, or as you can see in the upper left and lower left photos here, tufa at Mono Lake. And then at places like Walker Lake and Avery Lake up in Oregon, there's calcareous incrustations of rock in that shallow water region, so what limnologists or lake ecologists call the littoral region. So it's on this kind of substrate that they find a protected habitat, and that's a really important habitat feature. So the presence of and distribution of these rocky habitats, whatever kind they're made of, is a really important habitat feature for them to be able to protect themselves from wave action. If they're not protected from wave action, then they will be cast into the lakeshore, especially when they're in that vulnerable pupa stage where they can't move around. You'll get these big windrows of pupae accumulating along lake shores. That's actually a really good piece of evidence we have from the past in lakes, in particular like Mono Lake at much lower salinities, that in fact it was a much more productive environment, because there's actually photographs that document that kind of thing, and in fact one of them is right up in the upper right-hand corner. So this is from Avery Lake during a time in the early 1980s when the lake was at a fairly low salinity, below 50 grams per liter, below 5% salt. And right at the water's edge, you see the blackening. Those are the adult flies. And then right behind them to the left, you see that big pile of kind of brownish material. And those are the pupa stages that have been washed up on the shore. Not good to be washed up on the shore as a pupa stage because you're vulnerable to desiccation, you're vulnerable to predation by birds, and you're also vulnerable to parasitism. There's a small wasp that will often parasitize these pupae when they're up high and dry on the lake shore. So the two brine flies of most importance insofar as being a productive species of brine flies that are available as an important food resource to birds feeding at these different lakes in the Great Basin is the brine fly Ephedra gracilis that prefers chloride chemistry, and the alkali fly Ephedra hyans that prefers carbonate chemistry. These are both prominent in Great Basin lakes, in particular on the eastern side of the Great Basin in the case of gracilis, and in the western part of the Great Basin in the case of hyans, although they can be mixed. In fact, Ephedra hyans does occur at Great Salt Lake during lower salinity conditions. So in fact, perhaps under the conditions that the lake is undergoing right now, we may see Ephedra hyans be able to invade the lake once again and be present alongside Ephedra gracilis. Let's see. Again, they have specializations in water chemistry. But how do they cope with high salinity? So I'm going to dive a little bit in here into the weeds of physiology of flies and explain to you a little bit about how they do, in fact, cope with these high salinities. So alkali flies, brine flies that live in these high salinity salt lakes, much like us, they keep a concentration of blood salts at the same kind of concentration of blood salts we have in our own blood, at a relatively low concentration. Now, if they were isoosmotic, if they let their blood be at the same concentration of the water that they live in, they would sit along this dashed line called the isoosmotic line. So what you're seeing here is the concentration of osmotically active particles, that is to say dissolved minerals, dissolved salts along the x-axis, and along the y-axis would be the concentration of salts in their own blood. So if they were to follow, if their blood was to follow that same concentration along that isoosmotic line, they would be in trouble, because metabolically you really can't function very effectively if the concentration of your blood gets up into high salinity. So they have specialized physiological mechanisms known as osmoregulation for keeping the concentration of salts in their blood at a relatively low level. And this is an expensive process. It requires a lot of ATP, the energy currency of life, to keep those salts pumped out of their blood, and they have different specializations for those things in different species. And the osmoregulation line that you see here is about the concentration that they keep their blood at. In the case of the alkali fly, it's at about 300 milliosmolar. So as you increase salinity, now I'm going to show you the difference between what happens in the Great Salt Lake flies, Ephedra gracilis, and in the flies from Mono Lake, Ephedra hyans. So in this graph, once again, you see the dashed isoosmotic line and the Ephedra hyans osmoregulatory line, again at around 250 to 300 milliosmolar. But Ephedra gracilis, look at it, it's right up there between about 700 and 800, which means that in effect, the gradient between the internal concentration in its blood and the external concentrations of the lake water that it's living in is effectively reduced. So that means it's less expensive for Ephedra gracilis to live in salt lake water than it is for Ephedra hyans. So that means it can live in saltier water, and that's exactly what we see. So optimum conditions for salinity adaptation in the alkali fly are going to be much lower than they are for Ephedra gracilis. So for example, for the alkali fly, the sweet spot for them is right around 50 grams per liter, about 5% salt. Whereas for the brine fly from the Great Salt Lake, and I should consult with my colleague Wayne Wurzbeicher, who's done experiments on this kind of thing, my recollection is that it's somewhere up around between 100 and 120 grams per liter. In any event, at a higher salinity that they can tolerate, but not at such a high salinity that it's not without expense. So as the salinity increases, the expense increases, but the consequence of that is that they grow more slowly, and even though they grow more slowly, they don't achieve the same size of maturity that they would at a lower salinity. So as the salinities go up, it takes them longer to grow, they only achieve a smaller size at maturity, and the population growth rate is much lower, so the population density is smaller. So there's a real cost to this osmoregulatory adaptation that has its consequence of being a lower abundance and less food value per individual fly, and thus a less available food source to birds that are at these lakes that are undergoing higher salinity conditions. So just to bear that out a little bit more, and I think maybe I have a couple more minutes, I just want to show you an experiment that we did at Mono Lake. These are mesocosm tanks that were adjusted to different salinities of Mono Lake water, and to show you what the outcome of this experiment looks like. I think of these as kind of like time machines. So if you want to know what things were like at Mono Lake under past conditions, or what they might be like in the future, you can adjust the salinities to anticipate what the future salinity might have been for the lake, or what the conditions might have been like for the flies at a time in the past. And so this cartoon shows what the experiment did, what the outcomes of the experiment was, where we adjusted salinities between 50 grams per liter, which was the historic low salinity of the lake at the time. Anyway, water diversions began in 1941. And then we anticipated they might go as high as 150 grams per liter had not there been the decision by the water board made. Well, the experiment that we did at 150 grams per liter, no flies emerged at all. So what you can see from this is a series of experiments at 25 gram per liter increments, from 50 to 75, 100 to 125 grams per liter. And the cartoon is basically showing you that at 50 grams per liter, not only are there many more flies emerging, and there's much more algae growing that's available to them as a food resource, that the individual size of the flies is much bigger, and the slice of fat that makes up the body is much greater than at increasing salinities. So you can see from this experiment that it demonstrates the importance of salinity to productivity of the flies. I'll talk about that later and leave room for my colleagues.
So after talking to Dr. Herbst, we were tasked with monitoring these brine fly species at Great Salt Lake, out at the lake. And so I'm just going to kind of briefly go through the different methods that we went through. We had all of last year to play around with it, and we learned that a lot of the things that Dr. Herbst did in California didn't quite translate over to us here at Great Salt Lake. So there was a lot of trial and error trying to figure out what works and how to best get samples of. We were looking at the brine flies. A lot of it was Ephedra gracilis, and some of it was Ephedra hines as well. So we started off with, we would go out to the lake in a few different locations, and we would put out these cement blocks to try and mimic a microbialite and leave it out at the lake for 3 to 4 weeks, take it back to the lab, wash them, and count all of the larvae and pupa that were on them. So this is iteration 1. We call it the Atlantis hexapod, and it was hard to get it to go all the way into the sediment at the bottom of the lake, so we kind of scrapped that one and moved to a simpler version. So I don't remember if we had a name for this one, but we still were keeping the block. Oh, Excalibur, because it was like a sword. And so we would go out to our different sites, and this was last summer that we were putting these out. And then we took a bunch of groups out, and they helped us collect data. So this was a group of UES students last summer who were helping with our sampling. And then we've upgraded to the Medusa. We noticed that the blocks that were out at the lake were not growing enough of the algae on them to really attract the larvae. And the flies were also not pupating on the blocks. You would see, you walk over to collect your sample, and then everything would be getting kicked up from the sediment, but nothing would actually be on our blocks. But we did notice that the strings would collect a bunch of stuff on it, like the filamentous bacteria and just kind of debris from the lake that the flies would then pupate on. So we decided to just put a bunch of strings out there. And this one is actually out at just next to Great Salt Lake Marina State Park. So if you're out there and you see this, just leave it alone. And we've also kind of just decided to use nets and scoop up samples just from the bottom sediment of the lake and count them that way. We noticed that in 2022 in 23 when the microbialites were exposed, that a lot of the brine flies were just pupating on the bottom because there wasn't really other places to go. So we've been collecting a lot of samples that way. These are our sampling sites. We have one on either side of Ladyfinger Point and Antelope Island. We have one near Black Rock, one near on the other side, kind of where I showed that last picture of Great Salt Lake State Park Marina, and then 2 sites kind of narrowed down to one out at Gillmor Sanctuary that we had help with Heidi, who would go out there with us. And then we had another site out by not quite at the breach, but where the causeway is, and that site was a little harder to get to, but we got a couple of data points from that site as well. We tried to paddle out and get samples that were not just accessible on foot. It was a little hard to do, so we talked a lot with our colleagues about the shallow water samples being both easier to get to, but that's also where the flies are mostly at. And this is what our samples looked like. We collected a lot of them, and so we have last season's worth of samples, and just starting this season as well. And last year there was lots of flies. Yay! And just shout out to one of our graduating students, Cora Rasmussen, who did a lot of this work and a lot of those photos. Credited to her, has a poster that is displayed right now, so you should go talk to her about and get some more detail about these methods.
Hello, I'm Heidi, the floating head now, I guess. Anyway, so I'm the senior manager out at Gillmor Sanctuary, which is owned and managed by the National Audubon Society. And I want to talk about the sanctuary as part of the setting for the flies as a alternative habitat and a refuge, and particularly in light of food for thought. Well, we just happen to have the most awesome of these flies because they're the diving flies, as Dave described, where they gather air when they're adults and they can actually go underwater and forage and lay their eggs. And that is because we don't have as high of a salinity out of the sanctuary. We have a range of brackish to saline wetlands, that one of which goes up to and above 17%. And then, but we haven't yet found the gracilis, gracilis. And that is just because we haven't been looking as thoroughly, and I think that with these ephemeral wetlands that we do have as part of our system, we may end up finding them in the year or so. Anyway, why macroinvertebrates? Well, first of all, they're highly nutritious. They replenish energy reserves for the birds that come through and depend on them or stay and breed. And then there are a high percentage of the shorebird diet. And by the way, Gillmor Sanctuary is managed for shorebirds, although we do welcome other birds, I always tell everybody. So I came across this dissertation from Mark Nixon, who was a PhD student here at the U, and he was mostly interested, some of you in the back can't see the bottom lines here, but he was studying deer flies and their abundance, particularly out in the South Shore area. And he did his work out at the Baxter Duck Club, which is right next door to Gillmor Sanctuary, which is known for the world capital of deer flies. It happens that this fellow, this was not the focus of his research, but he was curious, like, well, who eats these? Who's the predators, the main predators? And so he was curious about shorebirds and took about 8 different species here. And while he was looking for the tabanids, who were in the deerfly, horsefly, large, painful biting bugs, which are starred with asterisks, he also found that most of these birds, all of them, either ate chironomids, which are the non-biting midges, our friends, and carixids, the water boatmen. And of the most opportunist, of course, is the American avocet that earned the Blue Star, next to number 7 birdshot, found in his gut. And not that it was shot, but it actually ate it. So of the birds that had the tabanids, only 3 of these species, the black-necked still, killdeer, and willet, actually ate the tabanids. Why is this important? Well, we know that the shorebirds are particularly interested in macroinvertebrates in their larval stage, typically. And we know that many of them love the chironomids, as mentioned before. But we have an opportunity to manage a number of different habitats for birds that cue in on different regimes of salinity and landscapes. Typically, the shorebirds like open landscapes, not the reeds and marshes that most of you are familiar with for what you might think a wetland is. And so in 2018, I decided to start to develop a monitoring protocol to better inform our habitat management decisions. In other words, how to grow irresistible bugs for the birds. And so we focused on the relationships between water management, because that's something that we could control, and the macroinvertebrate response. And at the same time, I took measures of biological, physical, and water quality parameters, and at sites foraged by the birds, shorebirds. And these are just some of the metrics that I developed at the time. We do a bird survey upon arrival, and then we collect these other things, which we don't need to go over right now. After a couple of years, we found that we had over 40 taxa, and I'll put that thus far because, as I said before, we weren't particularly seeking out, the extent of taxa that we have. We were mostly just trying to see what was available to the birds where they were foraging. So now we have this other added interest of, when we experience lake levels as low as we did in 2022, where's the refuge at? And how can we better manage? So that's always a possibility for not only the fly populations, but for birds as food. So you'll see these are all the usual characters we find out there. And we hear a lot about Wilson's foul ropes. These are redneck foul ropes. And one day when I was sampling all day, we had upwards of 5,000 of them out in this one pond. And it wasn't until about 5:30 in the afternoon that they dared come near me, and it's just only a few of them. They're very skittish. And so it was just really cool to be sampling the water column and the sediment that they would whirl around to try to bring invertebrates up to the surface. And also to just spend some time with them and observe their feeding patterns and knowing that they're picking from the water surface mostly. They likely were feeding on emerging adults from the various species that we have in this pond. This is a more freshwater pond, and this happened in two consecutive years, 2019, 2020, and they haven't come back in those numbers since, but it just makes me feel that this is a pelagic bird, typically, and not a lot is known about what they do on their way down to the Pacific Ocean during the wintering months. And we do provide forage for unsuspected species. So we're working more on that question about what it is, what they're seeking, and why, if there isn't adequate habitat out of the lake, and there's a great source of food over in this great place called Gillmor Sanctuary and other wetlands that are managed around the lake, perhaps we can help them. So as long as there's water, and please remember this, we can provide diverse habitat settings that maximize macroinvertebrate productivity. But we really need to focus on the resilience that this provides towards future threats related to climate change and water development. I should have thrown in there. But, and that's just to a certain threshold, of course. So in a northern temperate climate, you can expect that multiple generations will happen within many of these species of macroinvertebrates that we sample and observe. And those could increase to 4 or 5 or more generations, meaning that there's a longer period of time for the birds to come and forage if they arrive early, which they actually are. And then of the macroinvertebrate species, the highly adapted and tolerant species are the ones that are going to suit these opportunistic foraging shorebirds, and those such as the chironomids tend to be one of those most tolerant. So a lot of the macroinvertebrates that the limnologists use for identifying pure water versus not so clean water, and the more tolerant species are very parallel with the ones that are more tolerant of higher salinities. And then lastly, during periods of drought, it's also important to note that there's taxa that can relocate via flight, and those are these trichochryxids, a subgroup of or related to the chryxids, and then others that are tolerant of desiccation, meaning drying up periods via diapause. So they literally take a pause in their life stage and wait for more water. So lastly, I just want to put out there that we do have the tools to promote resiliency in terms of a habitat specific for the needs of shorebirds. And we do this by sharing what we learn in collaborative studies, such as the one I'm involved in right now with these wonderful people. And think in terms of managing the resources we have as a refugia for years to come, because the birds are counting on us to get it right before it's too late. So
Just so that I'm not shouting at everything. So, very nice to be here. Thank you all so much for having me. My name is Amanda Barth. I am the Rare Insect Conservation Coordinator for Utah State University, and I am going to discuss a little bit about brine flies as a conservation priority for the state, as well as how state conservation planning is going to include more of a focus on the Great Salt Lake. So, I would love that. I need to be on script, otherwise I forget.
Okay,
Oh, yeah.
so
Yeah, you can't hear me if I'm good at projecting. Okay, so, as a little bit of a background for the program that I run, I established Utah's Rare Insect Conservation Program in 2019, essentially from scratch, because nothing like this program existed in Utah before, and it also really doesn't exist in any other state. So this involved defining what our objectives were going to be and what the most efficient way to use our time and resources was. So our primary objectives were, we've defined our primary objectives as identifying conservation priorities by gathering available data for presumably at-risk species and their ranges, determining population trends at whatever scale possible. There's not always data to inform those things, and evaluating the scope and severity of threats. And so these factors allow us to calculate a given species' state-level conservation status, which we refer to as an S rank. If you've ever been involved in NatureServe, or you've looked at NatureServe's Explorer, they give you those values as global, national, or state-level ranks, and that's what we're looking for, a state rank. So we also compile information on the ecology of our focal species to better understand their habitat needs and their ecological roles, and we organize all this information into the Rare Insect Database, which we are also developing as through the Natural Heritage Program, the state's Natural Heritage Program. And specifically, we work to, specifically, I scanned too far. Sorry, notes. So our program is just 2 people. It's me and my technician, and we rely extensively on partnerships with federal and state agencies, with academic institutions, with nonprofits, with community organizations, with just researchers, and community groups that help us develop this database and identify recovery goals that define appropriate conservation actions and restoration and management priorities and objectives, and ways to mitigate threats that we've identified. We understand the scopes, we find the edges of everything, and then we try to tackle it. And specifically, we work to prevent species listings under the Endangered Species Act, which come with a suite of federal regulations and restrictions and permitting, and so that's against the state's interests. So it's a lot easier and a lot smoother for conservation before there's a listing than as a response to a listing. So, our program has worked to define which insect species meet the criteria to be considered species of greatest conservation need. And there's a lot of acronyms, I hope you brace yourself for this, SGCNs. And this is kind of a standardized terminology that's shared across all the US states and territories. And this is a designation that reflects the state's conservation priorities for at-risk wildlife and the habitats that they depend on. So species of greatest conservation need are identified and compiled across taxa groups. They guide conservation and restoration needs to support vulnerable species and are informative when defining key habitats and biological communities where actions would be most beneficial. So key habitats are another terminology. This is a term that we really rely on in our conservation planning. So importantly, because SGCNs are identified and described using this common terminology and criteria, states can then demonstrate the need for federal funding to support these conservation priorities and support these actions. So there's grants that are available specifically for species identified as SGCNs. And that means that there's going to be a compromise. There's got to be some balance between taxonomic representation and competition for these limited resources, or just finite funding. Since 2005, as part of the National Conservation Agenda that was laid out by the U.S. Congress, each state and U.S. territory developed a formalized wildlife action plan. We call them state wildlife action plans, or SWAPs, it's another acronym. And these were meant to promote recovery and viability of at-risk species, to prevent federal listing, or to meet recovery targets to delist or downlist species that have to be listed, wherever possible. So the conservation strategies in Utah's Wildlife Action Plan are guided by our identified SGCN focal species. So we develop this whole list of SGCN focal species. We understand, we get a sense of their habitats and their needs and their threats, and then we develop conservation actions and strategies around that. So this helps drive and prioritize the development of a mutual wildlife action plan. So it's a statewide plan. It's meant to incorporate the needs and priorities of all of the land management partners in the state, and that could be federal, state, and private. And ideally, these strategies are proactive, and they are based on current science and optimal community dynamics. But realistically, especially with insects, we don't always have abundant data to work with, and so we're more defining our actions based on historically limited data and very few resources. We don't have management authority for insects in the state, which is why I work with USU. Expertise is also a limitation, and so we try to develop preemptive conservation strategies, but sometimes they are unresponsive. Sometimes they're more reactive. So because these are enormous plans that undergo comprehensive revisions on a 10-year cycle, at minimum it has to be 10 years, we're now working on the third iteration of this plan for the state. So the 2025 UWAP revision is underway right now, and I'm participating in it extensively. And it's really only the third version of this statewide conservation plan. And so we're trying to make it really meaningful and inclusive of partner priorities. So the best part is that we're including in this iteration insect conservation concerns, which in my mind seemed like a given, but it has not been done before. So this is a really exciting new development, because that means key habitats that support those insects are also going to be included in the State Wildlife Action Plan. That's a really big deal. If you guys don't recognize what a big deal that is, it's a big deal. It's a big deal. I'm super excited about it. And that means a lot of vulnerable insects that we don't already even have data for might be protected by actions that are benefiting insects we do have data for, as well as other species that rely on those dynamics. So this also enables a much more genuine landscape level, a landscape level conservation approach. And if we're benefiting insect diversity, we're benefiting ecosystem function. And it's a lot more efficient and sustainable to target insects at a community level and treat them as both habitat and as wildlife. Habitat and wildlife, because they occupy a really essential point in the trophic levels of most of these terrestrial and aquatic ecosystems. And this is a perspective that was absent from the past 20 years of conservation planning. So I think it's a big point. It's a big point that I want to send home to you. So because this is the first UWAP revision that will include insects, we had to make a few adjustments to the criteria that were including insects as species of greatest conservation need. And most notably, we're accounting for the outsized role, the outsized ecological role that some insects play as keystone species. So brine flies really are a keystone species. They're not facing an extinction risk right now, but they are demonstrably essential to the function of the Great Salt Lake ecosystem. At their peak size, each year they are estimated to be, the adult numbers are estimated to be in the billions. And this makes them an incredibly important food source for millions of shorebirds and invertebrate predators like spiders and beetles. Their huge population numbers and metabolic success, as you heard from Dr. Herbst, also rely on the lake's salinity to be within a suitable range, meaning that the Great Salt Lake levels must be above a certain elevation to support billions of healthy brine flies, and in turn, the wildlife that prey on them. I'm not keeping up with my clicking and my notes here. Oh, so another really exciting development in addition to this 2025 UWAP revision is the inclusion, this is a big one, drum roll, the Great Salt Lake ecosystem is going to be included as a key habitat in this 10-year revision. It's a huge deal. This will address countless ways that this ecosystem supports and relies on the diverse biological community that use its resources, as well as taking into account a lot of climate resiliency factors and goals. We have a lot of targets that we're establishing involving climate and projections. We know that the Great Salt Lake serves as breeding grounds and migratory refugia for an estimated 10 million or so migratory birds each year, and supports more than 330 species of birds, and including the recently petitioned Wilson's phalarope. So the Wilson's phalarope relies heavily on a diet of brine flies. I'm repeating a lot of what you've already heard, and brine shrimp while it breeds in the Great Salt Lake. And this is another thing that helps highlight the importance of the Great Salt Lake. So the surrounding wetlands are also biodiversity hotspots and support dozens of mammal species, reptiles, fish, invertebrate species like insects and spiders, and notably, the wetlands are also an important summer breeding habitat for monarch butterflies, which face a possible listing decision this December. So I have a lot of work to do. And so collaborative actions are really the heart of how my program is even able to interface with a lot of community and nonprofit and federal and academic partners and the state wildlife agency. So the brine flies do qualify as a species of greatest conservation need. We still need an S rank for them. And we need an S rank. The way we accomplish that is by having population trend information and targets for, and getting a sense of the scope and impact of threats. So there's some specific metrics that we still need data on. And this is, brine fly, the Ephedra bacillus, does not even have you can't search it in the natural heritage like large-scale database to nature serve. It's not a taxon that's included in there. So part of my work soon will be to request, submit a ticket, add that taxon to the nature database, and then we can start recording more occurrence records and threats and so on. So this is the beginning. We're really very much at the beginning. We're starting from scratch here. So determining that state rank also requires a great deal of capacity and expertise that I do not have in my small two-person program. But we are incredibly fortunate to have been working with the Great Salt Lake Institute and Drs. Baxter, Biedul, and Herbst, who have been developing these metrics and protocols. And so their contribution of their scientific understanding and their monitoring protocols and connecting that with the salinity work by USGS and everyone else, this is where we can start developing a clear picture of the targets and conservation actions that should be included and the language that should be included in the Utah Wildlife Action Plan. So we absolutely need to continue the population monitoring because we need to have targets for it. So brine fly conservation will also rely on cooperation from stakeholder agencies who manage land and water resources around the lake. And finally, because the Wildlife Action Plan is a statewide conservation guide, it will be more successful when community support and stewardship of our unique and finite natural resources are a priority. So there is still time, and this is where I want to encourage you to maybe take a picture if you're interested. Please contact Paul Thompson. He's the lead on the update, the revision for the Utah Wildlife Action Plan. And there is still time to provide input, especially on brine fly or Great Salt Lake management. If you want to contribute your perspective or your knowledge into this revision process, I encourage you to do so. Thank you very much. Thank you for letting me stumble my way through this.
Okay, wonderful. I have a little fly friend who's going to buzz around the room and help you with a microphone to address some questions. We would love to spend some time chatting for our next little bit. And she looks a lot like Cora Rasmussen as our team fly expert, and also as a poster. Please raise your hand if you have a question. Oh, someone does. Oh.
Can you guys hear me?
Yes?
So I think you're saying the brine fly bars are like the littoral zone, the shallow water parts of the lake. Now, what happens to the brine fly bar repopulation dynamics when we artificially raise and lower the lake? That's what
So your question is, well, yes, the preferred habitat for the flies is in the shallow water littoral zone. That's where they are the most abundant. If the lake level goes up or down, part of their response is going to depend on the availability of the rocky habitat and microbialites in the case of Great Salt Lake. So I think a careful mapping of the availability of that substrate is going to inform a lot of what we will understand about how the population may respond. But that will also be conditioned by what the salinity is. So both the salinity level and the availability of that rocky intertidal environment will be critical to determining the success of the flies.
Yeah, Hannah in the back here. Oh, yeah, Wayne here.
Just to comment on that, we did population monitoring using scuba gear a number of years back on the brine flies, and we didn't find any density differences between, I think it was 0.5 meters and 3 meters of depth. But as Dave said, well, when we were monitoring, we were only sampling on microbialites. So that's a caveat on that. But you know, they were spread fairly deeply, and we didn't sample deeper than that.
Is there any evidence for spatial heterogeneity of where the brine flies like to live? Like, are there certain areas of microbialites where they're preferably found, or is it kind of homogeneous across the south arm of the lake?
The abundance of the flies at different locations will probably be a function of what local salinity conditions might be as well. But if that's uniform, then I'd expect them to be able to inhabit at similar densities anywhere around the lake on that rocky substrate. It's the presence of that rocky substrate that really determines where they're going to be their most abundant.
Carly, do you want to differentiate the sites that you saw in terms of how different it was, like at Lakeside, for example, than at Black Rock and in Antelope Island? What kind of variables at sites might play spatially?
The biggest variable was just wave action, given that we were in spaces that were getting a lot of the winds and the storm. So I think that was the biggest difference, but it was pretty uniform across different sites, and the way that we were sampling, we were getting very similar numbers, even though our data was not completely conclusive, but we were observationally getting really similar numbers across the entire South Arm.
Hey, I'd just like to know if the rock berm or the causeway itself is good brine-fly habitat, or if the presence of that causeway limits fly habitat on the north side.
So we didn't actually sample specifically on the causeway substrates, but the causeway substrates, when I've looked at them, they don't have that same kind of calcareous texture as the habitats elsewhere, where there's actually much more of the microbialite formation. I suspect if that rock substrate stays there for long enough, it will eventually form the microbialites on it and then become a favorable kind of environment. So now that you mention it, it seems like a conceivable way to create habitat, as has been done like in coral reef environments, is actually to place substrates out there and allow them to be colonized. So I hadn't thought about that before, but that's certainly a possibility to improve and increase the availability of habitat. Work on this one, please.
Just a quick question. I'm just wondering about the microbialites and if we are able to raise the levels of the lake again and they're able to be covered again, are the microbialites able to recover and how long does that take?
Oh, Carrie's right in front of you. Carrie really ought to answer that question. Cora, can you give Carrie France the microphone?
So,
Yeah. They're supposed to be better eyes.
the short answer is that we're doing that experiment right now as a lake rebounds, and we're going to keep an eye on it. But preliminary results suggest that there is a partial recovery, but it's slow and the community feedback is different.
Same as in our lab, the community is very different that starts growing on these dried out microbialites when we have them in tanks. And it's super interesting. It's like succession in ecology. So there's like pioneer species, and Olivia's back there, and that's what she told me. She's my student. Yeah. Okay, someone, oh yes, Dan.
Thank you for your talk. I was going with Flyman, so I like Lord of the Flies a lot better. But appreciate you being here. You mentioned the 3 instars and then the bubble that gets it up to the. What kind of studies have been done on that bubble and the fragility of the ejection? I'm just kind of curious.
Sure, yeah.
Or if
So-
that could even be studied. Yeah.
Yeah, it can be. So there are 3 larval instars, and at the end of the 3rd larval instar, they pupate, they form that puparium, and that puparium effectively isolates them from the outside water. So what's happening inside that puparium is that all the larval tissues are being converted into adult tissues. That means there's a lot of metabolic gases that are being generated. So the bubble of air that they're in is essentially the metabolic gases that they've been generating over the period of time that they've been developing into an adult fly. So when they crack off this little hatch cover, which they do so by blowing up what looks like a little balloon inside their head, and it pops open the hatch cover on the front of the pupa. They crawl out of that, but once they've freed themselves of the pupa, it's the bubble of air, metabolic gas that they're enclosed in, that floats them up to the water surface, and then that pops when they hit the surface. Yes, I've studied that. And our time is up.
Thank you, Team Fly.
I'd like